Welcome to the deep dive. Today, we're really getting into the miniature universe of microbes.
We are. We've got this great overview of key concepts here. And yeah, the plan is to pull out the most important, the most fascinating bits for you.
Right. Quickly and thoroughly, that's the aim. Because there's just so much variety, isn't there? Bacteria, viruses, fungi.
And even things like prions as the cellular ones.
Hmm.
So yeah, think of this as your essential guide to the basics of this hidden world.
A world that's been around for billions of years. I mean, these tiny things have ancient roots, and they've shaped everything, our health, the environment.
Totally, silently running things behind the scenes. So where should we begin? Maybe the germ theory.
Good starting point. It's kind of wild to think that before that, people blamed diseases on, what, bad air, miasma?
Exactly, or divine punishment, things like that. The germ theory was revolutionary.
Just the basic idea that microbes cause disease. It changed everything. Medicine, public health,
Yeah.
how we even think about being sick.
Absolutely. A complete paradigm shift. And thinking about how these microbes work, let's take bacteria. How do they, interact with stuff? Attach, move.
Ah, right. They have specific tools for that.
They do. Like Fimbria, you can think of them as tiny little Velcro hooks.
Velcro, seriously?
Sort of. They help bacteria stick firmly to surfaces, even in flowing liquids. Like the bacteria causing gonorrhea use them to attach inside the body. It's crucial for them.
Wow, microscopic Velcro. Okay. And then there are pili and flagella for moving around.
Right. Flagella are the well-known whip-like tails for swimming. Pilia can also be involved in a type of movement, kind of like grappling hooks, and also attachment. And this movement isn't random. They often move in response to signals.
Signals like what?
Well, like light, for example. That's called phototaxis.
So they move towards light or away from it?
Both,
Wow.
potentially. Some bacteria use light for energy, so they move towards it. Others might find intense light harmful, so they move away. It's all about finding the best spot to survive.
Driven by pigments that sense the light, I suppose.
Exactly.
Yeah.
Simple, yet effective sensing and response. It's amazing how adaptable they are.
Adaptable, definitely. And speaking of survival, endospores, these things are just incredible.
Oh, they really are.
Yeah.
They're not for making more bacteria, not reproduction. They're purely protective.
Like a panic room for the bacterium.
Kind of, yeah. When conditions get really tough, super salty, boiling hot, freezing cold, radiation, no food, the bacterium forms this incredibly tough, dormant structure inside itself.
And the rest of the cell just goes away.
Pretty much disintegrates, yeah. Leaving behind just this ultra-resistant endospore containing the essential DNA and components. And these things can last, well, potentially for ages.
Ages, like hundreds of years.
Try hundreds of millions of years, maybe even longer based on some findings. Just waiting.
That's absolutely mind-blowing. 250 million years dormant.
And then if conditions improve, boom, it can reactivate and become a normal bacterium again. Masters of survival.
Unbelievable. Okay, let's shift gears slightly. The basic types of cells, prokaryotes versus eukaryotes. What's the key difference we need to remember?
The biggest one is the nucleus. Eukaryotes, that's algae, fungi, protozoa. Us have a true nucleus where their DNA is kept wrapped in a membrane. Prokaryotes, bacteria, and archaea don't. Their DNA is just sort of free in the cytoplasm.
Simpler setup.
Generally, yes. Another difference is in the cell membrane. Eukaryotic membranes have sterols in them, like cholesterol in our cells or ergosterol in fungi.
What do the sterols do?
They add stability and flexibility. Think of them like little reinforcing rods within the membrane structure. Prokaryotes generally don't have them.
Okay, sterols for eukaryotic stability. Now, you mentioned gram-positive bacteria earlier. Something about titoic acid, where does that fit?
Ah, right, titoic acid. It's found in the cell walls of gram-positive bacteria only. Remember, they had that really thick layer of peptidoglycan.
Yeah, the stuff the gram stain detects.
Exactly. Titoic acids are these negatively charged molecules woven into that thick peptidoglycan layer. They help with structure, maybe attachment, kind of give the surface some of its properties.
Got it. Embedded in the thick wall, negative charge. Okay. Now, genetics. Bacteria have their main chromosome, but also plasmids? What are those?
Plasmids are extra bits of DNA, usually small, circular molecules separate from the main chromosome.
Extra, so not essential.
Not for basic survival, no. But they carry nice-to-have genes, things like antibiotic resistance, or the ability to break down unusual food sources. Bonus features, basically.
Like apps for bacteria.
Huh, yeah, kind of like that. And viruses. Their genetic situation is even wilder.
How so?
Well, unlike cells, which always use double-stranded DNA, viruses can have genomes made of DNA or RNA. And that DNA or RNA can be single-stranded or double-stranded, all sorts of combinations. Plus, the number of genes varies hugely between different viruses.
Very flexible, genetically speaking. Let's go back to that bacterial cell wall. Peptidoglycan. You said it's key.
Absolutely fundamental. It's this unique, mesh-like molecule made of sugars and amino acids that forms a strong layer around the bacterium.
What does it actually do?
Provides shape, structural support, and crucially, protects the cell from bursting due to osmotic pressure water rushing in. It's part of the cell envelope, which includes the cell wall and the cell membrane underneath.
And this is the basis for the Gram stain, the difference in peptidoglycan.
Precisely. Gram-positive bacteria have a very thick layer of peptidoglycan, plus those ditroic acids we mentioned. This thick layer traps the crystal violet dye used in the stain, making them look purple or blue.
Okay. And gram-negative?
Gram-negative bacteria have a much thinner peptidoglycan layer, and importantly, they have an extra outer membrane outside of that thin layer. This outer membrane has things like lipopolysaccharides, LPS, and porin channels.
So the thin peptidoglycan doesn't hold the purple dye?
Exactly. It gets washed out during the decolorizing step, and then they pick up the pink counterstain, safranin. So thick wall, purple, gram-positive, thin wall plus outer membrane, pink, gram-negative.
That's a neat way to remember it. And bacteria aren't just spheres and rods, are they? There's a whole zoo of shapes.
Oh, absolutely. You've got your Kochi, spheres, and bacilli rods. Then Cucurbitaceae short, plump rods. Vibrios look like commas, curved rods.
Spirals too?
Yep. Spirelles are rigid spirals, while spearshets are flexible, corkscrew-shaped ones that move in a unique way. And they group together differently too.
Like pairs and chains.
Right. Diplo for pairs, strepto for chains, staphylo for those grape-like clusters you see with staphylococcus, but also tetrads, groups of 4, sorcina, cubes of 8, even palisades where rods line up side by side.
Wow, a lot more complex than I realized. Okay, shifting to viruses again. They can't move themselves. How do they get into our cells? They need a way to attach, right?
Exactly. Enveloped viruses, the ones with an outer membrane layer, have spikes. These are proteins sticking out that are shaped to bind very specifically to receptor molecules on the surface of our cells.
Like a key fitting into a lock.
Precisely. It's a highly specific recognition and docking process. That's the first step, absorption.
Adsorption, okay. What happens next for animal viruses? Take us through the steps.
Okay, so after adsorption, binding to the cell, comes penetration. The virus, or at least its genetic material, has to get inside. This can happen through the cell engulfing the virus, endocytosis, or the viral envelope fusing with the cell membrane.
Then it has to unpack.
Right, uncoating.
About coating.
The protective protein coat, the capsid, comes apart, releasing the viral DNA or RNA. Then comes the really insidious part, synthesis.
Hijacking the cell.
Totally. The virus uses the cell's own machinery, its ribosomes, enzymes, energy, to make copies of its own genetic material and build its proteins.
And then puts the pieces together.
That's assembly, or maturation. New viral genomes get packaged into new capsids. Finally, release. The new viruses have to get out to infect more cells.
How do they get out?
Enveloped viruses often bud out,
Yeah.
taking a piece of the host cell membrane with them as their envelope. Non-enveloped viruses usually just cause the cell to burst open, lysis, releasing all the new viruses at once.
A hostile takeover, basically.
Right.
Now, cells themselves have internal structure, right?
Yeah.
Cytoskeleton. Even bacteria.
Yes, both do, though it's more complex in eukaryotes. In prokaryotes, they have proteins that act like a cytoskeleton. MIR-B helps give rod-shaped bacteria their shape. FTSZ is crucial for cell division, forming a ring that pinches the cell in two. Caryas helps make curved shapes.
And in eukaryotes, like our cells.
It's a much more elaborate network made of microfilaments, intermediate filaments, and microtubules. It provides support, helps the cell move, organizes things internally, transports stuff around, and is absolutely essential for cell division, mitosis, and meiosis.
A dynamic internal scaffolding. Okay, back to identifying microbes. We talked Gram stain. Are there other types of stains?
Yeah, broadly you have simple stains and differential stains. Simple stains just use one dye to make the microbes visible, see their shape and arrangement.
And differential.
Differential stains use multiple dyes to tell different types of bacteria apart or highlight specific structures. Gram stain is the classic example. Others include the acid-fast stain for bacteria like mycobacterium that cause TB, or endospore stains to see those survival pods we talked about.
Got it. Simple for basic shape, differential for more detail. Now, controlling microbes. Pasteurization, that's just for milk, right?
Mostly associated with milk and juices, yes.
Yeah.
Named after Louis Pasteur. It's specific heating and cooling to kill most harmful microbes without ruining the product. It's not sterilization, though.
What's the difference? Sterilization sounds more final?
It is. Sterilization aims to kill everything, including those tough endospores. Think autoclaving high-pressure steam. Sanitization is more about reducing microbial numbers to a safe level, like on restaurant surfaces. And remember, none of these necessarily deal with the cellular things like viruses or prions easily.
Right, those need different approaches. And even between bacteria and our cells, there are exploitable differences, like ribosomes.
Exactly. Bacterial ribosomes are smaller 70S type. Ours, eukaryotic ribosomes, are larger 80S. That size difference is a key target for many antibiotics.
How so?
Drugs can be designed to mess up the 70S ribosome function, stopping bacteria from making proteins, but leave our 80S ribosomes alone. Selective toxicity.
Clever. Okay, viral infections. Sometimes you get sick fast and get over it. Sometimes it lingers or comes back.
Right. We classify those timelines. Acute infections are rapid onset, you feel sick, immune system clears it, it's gone. Like the common cold or flu.
And the ones that come back?
Those are latent infections. The virus goes dormant after the initial illness. It hides in your cells, not actively replicating, but it's still there. Then later it can reactivate. Think herpes viruses, cold sores, shingles,
And the lingering ones.
persistent or chronic infections. The virus isn't cleared, it sticks around long term, often replicating at low levels. HIV is a key example, or chronic hepatitis B or C.
Acute, latent, chronic. Got it. What about fungi? They look different, often fuzzy.
Yeah, many fungi grow as these long, thread-like filaments called hyphae. A whole network of hyphae makes up the main body, the mycelium. That's the fuzzy mold you see.
And viruses have that protein coat, the capsid. What's it made of?
It's built from individual protein subunits called capsomeres. They self-assemble like little building blocks to form the complete capsid structure around the genetic material.
Hyphae and capsomeres. Okay. So how do labs actually figure out which microbe is causing an infection?
Several ways. Biochemical testing is a classic method. Grow the microbe and see what sugars it eats, what waste it produces. Kind of like a metabolic fingerprint.
And newer methods.
Genetic analysis is huge now. Sequencing the microbe's DNA or RNA gives a definitive ID. Techniques like PCR let you amplify tiny amounts of microbial DNA to detectable levels.
Looking at their genes directly. Okay, circle back to prions. You said they're just misfolded proteins. How do they cause disease? It sounds bizarre.
It is bizarre and pretty scary. A prion is an abnormally folded version of a protein that's naturally present, especially in the brain. The scary part is, this misfolded prion protein can somehow induce normal copies of the protein to misfold in the same way.
A chain reaction of misfolding.
Exactly. These misfolded proteins clump together, accumulate, damage brain cells, and lead to devastating neurodegenerative diseases. The brain tissue ends up looking spongy, and currently they're fatal. No cure.
Truly chilling. Okay, something slightly less terrifying maybe. Bacteriophages. Viruses that infect bacteria. How do they affect us?
Indirectly. They don't infect our cells, but they can change the bacteria they infect. Sometimes a phage transfers genes to a bacterium that make the bacterium more harmful to humans, like giving it the code for a toxin.
So the virus makes the bacterium more dangerous. Wow. Okay, big picture. Oxygen, we need it to breathe. Where does most of it come from? Trees?
Trees are important, absolutely,
Yeah.
but the heavyweight champions of oxygen production are actually algae.
Algae, like pond scum.
Well, yeah, but also microscopic algae in the oceans, phytoplankton. They perform a huge amount of the planet's photosynthesis, taking CO2 and releasing the oxygen we breathe.
Yeah.
Unsung heroes.
Definitely. Now, microbes often live in communities, right? Biofilms.
Yes, biofilms are a huge deal. They're communities of microbes stuck to a surface, living together inside a slimy matrix they secrete.
Where do we find these?
Pretty much anywhere moist. Dental plaque is a biofilm. They form on medical devices like catheters or implants, in the lungs in cystic fibrosis patients, urinary tract.
And they're hard to get rid of.
Very. The matrix protects them from antibiotics, disinfectants, even our immune cells. Plus, living close together makes it easy for them to share genes, like antibiotic resistance genes. They're a major challenge in medicine.
Fortified microbial cities. And some viruses can actually cause cancer.
Yes, certain viruses, called boncoviruses, can trigger cancer. They interfere with the cell's normal control mechanisms, pushing it towards uncontrolled growth. HPV causing cervical cancer is a well-known example, or hepatitis B and C linked to liver cancer.
So viruses can be pathogens, gene donors, and even cancer agents. Incredible range. We should probably mention some pioneers here, the people who figured all this out.
Absolutely. Couldn't have this conversation without them. Semmelweis pushing hand washing in hospitals.
Lister and sterile surgery.
Jenner and the first vaccine for smallpox.
Yeah.
Pasteur, obviously with pasteurization and really cementing the germ theory.
And the microscope guys. Hook seeing cells, Leeuwenhoek seeing the actual microbes, the animalcules.
Couldn't see any of this without them. Foundational work.
Okay, back to bacteria. We mentioned pili for sticking and moving. Don't they also share genes?
Yes, that's another key role for some types of pili, often called sex pili. They facilitate conjugation.
Conjugation.
It's direct transfer of DNA, usually plasmid DNA, from one bacterium to another through a pilous bridge, a major way antibiotic resistance spreads.
Direct gene swapping. And some bacteria need specific nutrients they can't make, growth factors.
Right. Essential organic molecules, vitamins, amino acids that they have to get from their environment. If they need a lot of these, we call them fastidious.
Picky eaters. So conjugation is one way to get genes. What are others?
There's transformation, picking up naked DNA released from dead bacteria. Transposition, where jumping genes move around within the genome or to a plasmid. And transduction, where a bacteriophage accidentally carries bacterial DNA from one host to the next.
Wow, multiple ways to mix and match genes. Keeps them adaptable. How do they actually reproduce? Split in half?
Pretty much. It's called binary fission. The cell duplicates its chromosome, the copies move apart, the cell gets longer, and then it divides in the middle. Makes two identical clones. Very efficient.
Simple and fast. And they have preferred temperatures, right? Like us, but more extreme.
Definitely. We classify them by temperature range. Psychrophiles love the cold, down below freezing even. Mesophiles like moderate temps, like our body temperature. Most human pathogens are mesophiles. And thermophiles love heat, hot springs, hydrothermal vents. Some even go way higher, hyperthermophiles.
Life finds a way, even in extreme heat or cold. Now, mistakes happen during DNA copying. Mutations.
Yep, changes in the DNA sequence. Point mutations affect just one base. Can be silent, no change of the protein. Missense, different amino acid, or nonsense, creates a stop signal, truncating the protein.
And frameshift.
That's when bases are inserted or deleted, but not in multiples of 3. It messes up the whole reading frame downstream, usually leading to a totally non-functional protein. Mutations can be bad, neutral, or sometimes even good for the microbe, like gaining antibiotic resistance.
Constant source of variation. And when bacteria grow in lab cultures, they follow a pattern.
A classic growth curve, yes. First, the lag phase, they're adjusting, getting ready. Then the log phase, exponential growth, doubling rapidly. Then stationary phase growth slows, deaths equal new cells, often due to limited nutrients or waste buildup. Finally, the death phase, more cells dying than dividing.
Predictable rise and fall. Let's define some core terms. Gene, enzyme, coenzyme, cofactor.
Okay, a gene is the DNA sequence coding for a product, usually a protein. Enzymes are proteins that catalyze biochemical reactions, make them happen faster. Coenzymes are organic helper molecules, often vitamins that assist enzymes. Cofactors are inorganic helpers, often metal ions that also help enzymes function.
Genes are the blueprint, enzymes do the work, coenzymes and cofactors help out.
Got it. And microbes interact in different ways.
Yeah,
symbiosis.
Right. Symbiotic means at least one partner needs the other. Mutualism, both benefit, like termites and their gut microbes. Commensalism, one benefits, other unaffected, like some skin bacteria. Parasitism, one benefits, one is harmed infections.
And non-symbiotic.
They don't strictly need each other. Synergism, they cooperate for a result neither could achieve alone. Antagonism, one harms the other, like producing antibiotics.
A whole social structure almost. And temperature limits, cardinal temperatures.
The minimum, optimum, and maximum temperatures for growth. Below minimum, membranes freeze up. Above maximum, enzymes fall apart. Denature. Optimum is where they grow fastest. Defines their comfort zone. Okay, energy. ATP is the goal. How do NADH and FADH2 fit in?
They're electron carriers. They pick up high-energy electrons during glucose breakdown and deliver them to the electron transport chain. As electrons move down the chain, energy is released to pump protons, creating a gradient that drives ATP synthesis. They're the link between food breakdown and ATP production.
Energy delivery trucks, and all these reactions together are metabolism.
Exactly. Metabolism is the sum of all chemical reactions in the cell. It eats enzymes, energy, nutrients. Metabolism breaks things down, releases energy. Anabolism builds things up, uses energy. Gene expression is turning the DNA blueprint into functional proteins, including those metabolic enzymes.
Building up, breaking down, making energy, the chemistry of life. Now,
Yeah.
For an infection to start, you need enough microbes, right? Infectious dose.
Yes. It's the minimum number of organisms needed to actually cause disease. Varies hugely between pathogens. Some need only a few, others need thousands.
A critical threshold. We talked about biofilms being tough. Why again?
The matrix blocks antibiotics and immune cells. The environment inside is different, slowing growth and making them less susceptible. And they easily share resistance genes. Really hard to treat infections on implants, for example.
A formidable defense. Besides heat, How else can we physically remove microbes? Filtration.
Yep. Passing liquid or air through filters with tiny pores traps the microbes. Good for heat-sensitive liquids or purifying air. Doesn't remove toxins, though, just the microbes themselves.
A physical sieve. Okay, our body's defenses. Innate immunity has weapons like defensins and interferons.
Right. Defensins are proteins on our skin and mucous membranes that poke holes in microbial membranes. Interferons are signaling proteins made when a cell gets infected by a virus. They warn nearby cells and interfere with viral replication.
Built-in antimicrobial systems. And drugs often target things unique to microbes.
That's the goal. Antifungals hit ergosterol in fungal membranes or chitin in their walls, things we don't have. Antivirals target steps specific to viral replication. Antihelminthics, for worms, block their unique metabolism or muscle function. Targeting eukaryotes like fungi and worms is trickier because their cells are more like ours, so side effects are a bigger concern.
Finding the differences. Okay, quick definitions. Contaminant, sterilization, sanitization.
Contaminant is just any unwanted microbe. Sterilization is killing everything, including endospores. Sanitization is reducing numbers to a safe level, not necessarily zero.
Different goals for different situations. Our immune system also learns, right? Adaptive immunity.
Yes, that's the specific targeted response. Develops after exposure, has memory, relies on B cells making antibodies, and T cells doing various jobs like killing infected cells or helping B cells. Slower to start, but gives long-lasting protection.
Innate is fast and general. Adaptive is slow, specific, and remembers. And antibiotics. Broad versus narrow spectrum.
Broad spectrum hits a wide range of bacteria, gram-pos and gram-neg. Narrow spectrum hits only specific types. You use broad when you don't know the exact cause, but narrow is better if you do, as it's less disruptive to your good bacteria. They target things like bacterial ribosomes, DNA synthesis, or cell wall building.
Different tools for different jobs. Fever and inflammation seem bad, but can be good.
Often, yes. Fever speeds up our metabolism and immune response and can slow down microbe growth. Inflammation walls off the infection site, brings immune cells in. Cytokines are key signaling molecules coordinating this.
Controlled responses to fight back. And phagocytosis, immune cells eating microbes.
Exactly. Cells like macrophages engulf bacteria, debris, whatever doesn't belong, and digest it. Key part of cleanup and defense.
Cellular Pac-Man. Antibodies are different types, IgA, IgG.
5 main classes. IgA is in secretions. Mucus, tears, milk. IgD on B cells. IgE involved in allergies and fighting worms. IgG is the main one in blood. Provides long-term immunity. Crosses the placenta. IgM is the first responder in an infection, good at clumping microbes and activating complement. They have different structures too, monomers, dimers, pentamers.
A specialized antibody toolkit. And complement, this cascade thing.
Yeah, it's a set of blood proteins that get activated in a cascade. Can be triggered by antibodies, classical pathway, microbial sugars, lectin, or just microbial surfaces, alternative.
Okay.
The end result, coating microbes for phagocytosis, opsonization, causing inflammation, and forming a membrane attack complex, or MEC. It punches holes directly into microbial membranes, bacteria, fungi, protozoa, causing them to fly and burst open. Very effective.
A whole punching defense system. Okay, decontamination again, just cleaning things up.
Pretty much. Removing or neutralizing unwanted microbes or hazardous materials.
And fever being helpful raises temperature to hinder microbes and boost our immunity.
That's the idea, yes.
Yeah.
Within limits, of course.
Right. Antibiotic resistance. How does natural selection cause it?
Simple but powerful. Antibiotic kills the susceptible bacteria. Any resistant ones survive. They multiply. Soon, the whole population is resistant. Survival of the fittest, driven by the antibiotic pressure.
Evolution happening right in front of us. Physical control methods, heat is big, dry versus moist.
Dry heat, ovens, incineration needs higher temps or longer times. Moist heat, boiling, pasteurization, steam, penetrates better. Autoclaving steam under pressure is the most effective moist heat method for sterilization. Kills everything, including endospores.
Autoclaving is the gold standard for sterilization then. And how do bacteria actually resist antibiotics? What tricks do they use?
They have quite a few. They can make enzymes to destroy the drug. They can stop the drug from getting into the cell. They can pump the drug back out using efflux pumps. They can change the target site so the drug can't bind anymore. Or they can develop alternative metabolic pathways to bypass the drug's action.
Clever defenses. Immunity. Natural versus artificial, active versus passive. Break that down.
Okay? Natural is through normal life. Artificial is medical intervention. Active is your body making its own response, long term. Passive is getting ready-made antibodies, short term.
So examples.
Natural active. getting sick and recovering. Natural passive, mother to baby via placenta or breast milk. Artificial active, vaccination. Artificial passive, getting an injection of antibodies, like gamma globulin.
Makes sense. And vaccines, artificial active immunity, how do they work at the core?
They expose your body to antigens, safe parts, or inactivated versions of a microbe. Your immune system learns to recognize these antigens, makes antibodies, especially IgG for long-term memory, and memory cells, all without causing the actual disease. Then, if you encounter the real microbe later, you're ready to fight it off quickly.
Training the immune system safely. And diagnostic tools like PCR, FISH, ELISA.
PCR amplifies DNA, finds tiny amounts of pathogen DNA. FH uses fluorescent probes to light up specific microbes or DNA sequences in samples. ELISA uses antibodies linked to enzymes to detect specific antigens or antibodies good for finding proteins.
Tools to detect the microbes or our response. Let's put some numbers or facts on specific microbes. Staphylococcus.
Yeah, serious issue. Staph infections cause something like 100,000 deaths a year globally. Not insignificant.
Wow. Measles. The vaccine debate.
Key point. Complications from getting measles naturally are way, way higher risk than any rare side effects from the vaccine. Pneumonia, encephalitis. It's a serious disease.
Vaccine is much safer. Flu symptoms.
You usually know it. Sudden fever, cough, sore throat, body aches, fatigue. The classic misery.
Yep. HIV versus AIDS.
Important distinction. HIV is the virus. AIDS is the late stage of the disease when the immune system is badly damaged. HIV infection doesn't automatically mean AIDS, especially with modern treatment.
Yeah.
And sadly, still no cure for HIV itself.
Treatment, yes, coronaviruses.
Big family. Cause anything from a common cold to severe stuff like COVID-19. Respiratory spread, wide range of outcomes.
That hepatitis A and B.
Good news there. We have effective vaccines for both Hep A and Hep B, crucial for preventing liver disease.
Vaccination works for those. Finally, Pseudomonas. Why is it tough?
It's naturally resistant to many disinfectants and antibiotics, forms biofilms easily, spreads in hospitals, needs really strict cleaning and sterilization protocols to keep it in check, especially around vulnerable patients.
A real challenge. Well, that was certainly a deep dive. We covered a massive amount of ground.
We really did, from the basic theory to specific structures, genetics, interactions, immunity, disease. It's a huge field.
Thinking about things like those ancient endospores, algae making our oxygen, the complexity of biofilms, it really changes how you see the world.
And hopefully you listening have picked up some solid fundamentals, had a few aha moments maybe, without feeling totally overwhelmed. It connects so many aspects of life and health.
Absolutely. It's a constantly evolving story too, isn't it? Microbes adapting, new challenges emerging, makes you wonder what the next big discovery will be or how our relationship with microbes will change in the future.
Always something new to learn.
Definitely something to ponder. Thanks so much for joining us on this deep dive.